Hello, folks. My name is Blaine Allen, and today we're going to be talking about mitosis. So, without further ado, let's get into this. So, here is our overview of plant mitosis. Allium is our genus name for our onion. So, we're going to be looking for, first of all, our interphase. So, it is not part of mitosis, it is part of the cell cycle. This is when the cell is spending most of its time. And what we're going to be looking for is that nice, neatly formed nucleus with a discrete edge and genetic material that is in the form of chromatin. It should appear grainy instead of appearing as little ropes. So real quick, let's go ahead and go to that on the microscope. All right, today we're going to be looking at slides that are a cross-section of an onion root tip. The reason why we have chosen an onion root tip as our model organism for this particular purpose is because at those root tips, we can expect to see a lot of mitosis going on. We also won't have to deal with chloroplasts, which we would have to deal with at the apical meristem, where plants would also be displaying growth, but it would be difficult to see the features of the nucleus. So, real quick, this is what they look like at 40x total magnification. And you can see we have two different onion root tips here on this slide. That's so that if we can't find what we're looking for on one, we can just go over to the other one and look for what we want there. And because these roots are growing downward, the most growth is going to be happening right here at the tip. So these are the areas where we're going to have the best luck finding cells undergoing mitosis. So let's go ahead and go up to a higher magnification. This is 100x total magnification. Already we can see some promising looking cells like that one, a few over here that are clearly in different stages of mitosis. So let me go ahead and go up to 400x total magnification. And here we go. Let me adjust the some of the settings a little bit. So I like this cell right here in the middle. I'm going to go ahead and put a drop of immersion oil on this onion root tip. And I'm going to go up to 1000x total magnification. All right. So this is the one I like. So this cell right here is a great example of our plant cell in interphase. So this is when it's just doing regular old cell stuff. So for an onion root, that would be storing some starches, transporting liquids and minerals up to the leaves, and receiving sugars from the leaves, that sort of business. So next, let's go back to our, before I go on, for this cell, what you want to label in your drawing, you can see that we have a cell wall out here. Know that you've got a cell membrane on the inside. But more importantly, what we're looking at for this phase is that you have a nucleus. You see that nice, neat edge on this. And on the inside, your DNA looks very grainy, and that's because it is in the form of chromatin, this uncondensed form that is ready to use for the cell. So moving on, let's go ahead and go back to our PowerPoint real quick. The next cell that we're going to be looking for is one in prophase. And in this phase of the cell cycle, the DNA is going to be in the form of chromosomes. Your nuclear envelope has dissolved, so we're no longer going to get that nice, neat edge on your ball of genetic material. Okay. Also, if you label a nucleus on your prophase drawing, you will get docked points because that is one of the key characteristics of prophase, is that the nucleus no longer exists. Let's go back to our slide and let's find a good prophase. I think I saw some pretty close by. Yeah. Yeah, I like this one the best. This is a good classic example of our prophase. So here is our prophase. You see our DNA is no longer grainy as in this cell right above it, but it has formed these little condensed chromosomes. That is what you're visualizing there. They're still kind of sort of in a ball, but that nuclear envelope is dissolving, and pretty soon they'll kind of be hanging all loosely within the cell. One thing you should remember is that this is a continuous process. We don't have these discrete steps. It's not like, it's kind of like child development. You don't just wake up and one day your baby's become a toddler. It's a step-by-step gradual process, and there's all sorts of in between. And so if we look over here, you might have noticed, we've got this cell where it's got DNA that's condensed, but it's still very nice and round. That's because it's just a little bit earlier on. And then you can find other ones that are a little bit later on, such as this cell right here. But back to this one. For your prophase, you will need to, in your drawing, address that there is no longer a nucleus, either by not drawing one, not labeling it, or stating it outright. And know that all you have is just a pile of chromosomes, just kind of hanging out in the blob where the nucleus dissolved. So imagine if you had a sack full of groceries and then your sack busted open. So they're just all kind of sitting on the ground, hanging out. You know what? I'm gonna, oh yeah, let me up the resolution at this point too. Go back to good prophase real quick as well. Okay. So back to the PowerPoint real quick. The next phase that we will be looking for is metaphase. It is not terribly easy to distinguish in the plant, I will admit. Metaphase is a lot more distinct in your animal cells, but we're going to go ahead and find some for you real quick. So let's go on the prowl for plant cell in metaphase. So let's just scroll around a little bit. That's a good one for later. Aha. Yeah, I know y'all think I was just being picky passing by all those ones that were kind of sort of right, but here we go. This is a nice textbook example of a plant cell in metaphase. You can see as I focus through it, those chromosomes appear to slightly change position, and that's because they're 3D structure and we're witnessing different planes of focus throughout it. Anyways, the important thing to note for this phase is the presence of these spindle fibers and the arrangement of your chromosomes. So your chromosomes are now aligned right down the equator of this cell. So right down the middle, we're about to pull them apart into the far corners of this cell. That way we can divide the cell into two new cells. So right here you see your line of chromosomes right down the middle and these little tiny fibers. You might notice you kind of see them all over the place in all of these cells, even the ones that aren't currently in the middle of mitosis. And that's the thing, your cells have this cytoskeleton. And the thing is that these centrioles that are creating spindle fibers, moving these chromosomes around, they not only serve this function in mitosis, but during normal everyday cellular function. If something needs to be moved around the cell, this is how your cell is going to do it. And that's why you see those same fibers in all these other cells, but only in these cells undergoing mitosis do you see this level of organization, because everything in the cell is all focused on getting through cellular reproduction so it can get back to the business of doing cell stuff. Okay, so this is your plant cell in mitosis. You have chromosomes right down the middle. I might draw those as little X's, and then spindle fibers attached to them, leading in a little kind of triangle shape to your centrioles out on this side and this side. Remember that your spindle fibers are always pulling your chromosomes apart. In the next phase, we will see a little bit of a gap in between these chromosomes. Your spindle fibers are not in between your chromosomes pushing. You can't shoot pool with a rope, okay? So remember, your fibers, they can pull, they aren't any good at pushing though. So let's go back to our PowerPoint real quick. So here is our plant cell metaphase. Now the next one we've got to look for is anaphase, when they are starting to come apart. And I remember seeing a pretty good one on one part of this root. So let's real quick go back to the microscope. Let's take a picture of that nice, good metaphase. And then let's go over here. And I know somewhere I saw a real good That's pretty good. Just hoping for one a little bit more. All right, here we go. Oh no, that one's kind of janky too. Aha, here's the one. Okay, so here's a great example of our plant cell in anaphase. You can see that now our chromosomes have separated. So our sister chromatids have separated. They are now daughter chromosomes. So at this point, you can start calling these daughter chromosomes. In that last phase, your sister chromatids were still attached. Once they're torn apart, they are now daughter chromosomes. So for your drawing of plant cell anaphase, you've got daughter chromosomes. They are still being pulled apart by spindle fibers. And the key feature here is that they're now no longer connected. They have been separated, but they're still very much in the process of being pulled apart. Let me find a nice, good looking plane focus. Yeah, that'll do. Snap a nice, pretty picture of that. So that is our plant cell anaphase. Let's go on over, and here we have one more phase of the cell cycle defined for plant cells. So let's go back to the PowerPoint real quick. So telophase, and I bet I can find a cell plate for y'all. I have the advantage of having a microscope with a 100x objective. So let's go up. Aha, bam! Just like I said. So here is our plant cell and telophase, sorry. So here, our chromosomes have separated. They are starting to form daughter nuclei. You can see very faintly here this cell plate forming. And this is very crucial. This is one of the crucial features that need to be in your drawing and labeled, because this is going to be different than the type of cytokinesis displayed by animal cells. So you have daughter nuclei, and you have a cell plate forming right there. Sound good? And this is our plant cell telophase and cytokinesis. Let me decide on a good looking plane of focus. That's pretty good. Let's snap a nice picture of it. There we go. Whoo! Good looking. All right. Okay, so that should cover it. Make sure you draw and label all the important features. And again, because we're talking about mitosis, the division of the nucleus and the genetic material, what's going to be important in each of those different steps is what form the DNA is in and what's happening to it. Where is it going? Yeah, so know that interphase, it's chromatin. Prophase, you have chromosomes. Metaphase, you have chromosomes still lined up along the equator. At this point, what you have is sister chromatids still attached to one another during prophase and metaphase. In anaphase, they separate. And they are now daughter chromosomes. And then once they start to reform nuclei, then they are daughter nuclei. And in your telophase, you're forming a cell plate. All right. If you have any questions, please do not hesitate to email your designated lab instructor. You all have a wonderful day.